So welcome to my presentation about the aromatic fingerprint of fermented Coffea liberica. I'm doing this as part of my PhD project, which is called Deep Authent. And the aim of this project is the development of a benchtop non-target screening approach for food and foodstuffs that is point-of-care usable. That means that we can transport the device that we are using to the point of care, so where either the farmers are growing the coffee or where the coffee is shipped to. And we want to do this for authentication of coffee, for quality analysis, and also for shelf life prediction. And in the future, we are going to use neural networks for data augmentation. That means we are measuring two authentic samples and then are more or less cutting those two together in silico, so on a computer more or less. But that's a little bit too advanced for today, so my presentation topic today will be the differentiation between Coffea arabica, canephora, and liberica, and also the differentiation of fermented and unfermented coffee. And in this specific case, it's fermented and unfermented Coffea liberica. And I did this using headspace GCIMS measurements of powdered green and roasted coffee beans. First, I want to go a little bit into some theory, which is why we are doing this. And as Raven showed the SCA grading charts and also the charts that Dr. Schwartz has, so what we want to do is somehow objectify these aroma impressions that are more or less subjective for everyone, because you have different ways that smell and what is often mistaken as taste comes to the olfactory receptors in your nose. That is one, the orthonasal, that is from the front of your nose, that is more the water-soluble aromas, and the retronasal way, so from the back of your throat while you're, for example, sipping coffee, which are more the fat-soluble aromas. And these receptors, or the number of receptors, is different more or less for everyone. And we have different perceptions also from what we learned in childhood, for example, as to what coffee tastes like. And we want to somewhat objectify that by using gas chromatography for aroma compound separation, where we can separate water-soluble and fat-soluble aromas in the same measurement and compare the proportion of the specific aroma, which is then a semi-quantitative method, but with a calibration we would also be able to quantify them. So a really, really short and tiny introduction as to how gas chromatography and ion mobility spectrometry work, which are the techniques that I'm coupling in the lab. So in gas chromatography, you have a capillary column onto which the, for example, liberica coffee is applied, or the gas phase of above the liberica sample is applied to, and then there is commonly a column oven outside of this column, which heats up the column slowly after the appliance of the sample. And so the compounds are separated by volatility. That means how fast or how high is the boiling point of these compounds. And also the interaction of the stationary phase, which is a coating along the outside wall of along the inside wall of this column, which has then interacts differently with differently polar molecules. So this is the first separation that I have in my instrument. Let's move from the left side to the right side, where you then can see the IMS. That is the detector that I've got at the end of my gas chromatograph. And there you can see that there is an ionization part in this cell, and there is a weak beta emitter that is ionizing the molecules that are coming from the gas chromatography column. And then those ions are drifting along an electrical field, and against this electrical field there is a drift gas, so in our case that's nitrogen, and by collision with those nitrogen molecules the ions are slowed down somewhat. So smaller ions that don't collide as much with the nitrogen molecules are faster at the Faraday plate and This means that this is a second separation-based technique based on molecule size and charge, and we are coupling those two techniques, one as the analytical side with the gas chromatograph and the detector side with an ion mobility spectrometer. And this is how, in theory, the spectra then look. This is important because I'm going to show you the differences in the spectra between the different coffee species and the ferments. You can see on the y-axis that this is the retention time, so the time that these molecules need to go through the gas chromatography column. And on the x-axis, there is the drift time, so the time that the ions then need to drift through the IMS cell. And the spectra that I show here, so the one on the left-hand side, that is the theoretical spectrum of a ketone mixture, and on the right-hand side here, you can see the actual ketone mixture, how it looks in my device. So what did I do with the coffee? I had 26 green coffee bean samples, which consisted of 8 Arabicas, 12 Canephorus, and 3 unfermented and 3 fermented Libericas. And I had 45 roast bean samples that were also consisting of 8 Arabicas, 12 Canephorus, but 8 unfermented and 17 fermented Libericas. I got them kindly from Dr. Schwartz. And what is necessary to say is that the Libericas were only from India, while the Arabicas and Canephorus came from the different farms that Dr. Schwartz is cooperating with. I then did headspace measurement. That means that I'm measuring the gas phase above the sample in this headspace vial and injected that into the GC-IMS system. And afterwards visualized the data with a Python tool that's called GC-IMS tools that my colleague and co-worker Joscha Christmann published last year. And also did the first step of our machine learning pipeline, which is a PCA that we did see a few times already during this day. So let's come to the differences between the different species. You can see that I marked 3 different boxes here, and that is to show you that those 3 species not only vary in general in taste, but you can also see that in the spectra. So we have this yellow box and the blue box here, and these show the differences in intensity in the spectra. So there are different intensities throughout the 3 species. And also we have differences in the occurrence of peaks, which is especially visible here with this red box, with this peak that can only be seen in Coffea arabica. And I then try to tentatively identify some of the compounds. The problem is that I did this by retention time index, and that means that it is not possible to have a look at the drift time. So this is just by gas chromatography, more or less. And therefore, I'm not able to really identify substances, but it's always a number of substances that are given that could be this specific substance. And for the roasted coffee, the substances that differed the most were mostly products of the roasting, so pyrrolidines, pyrrenes, pyrazines, and furfurols. For a real identification, we have to do compound elucidation by mass spectrometry and with a reference substance. I did the same thing for the green coffee. And what you can see firsthand is that the green coffee has significantly less signals in the IMS spectra, but still we can see differences in peak occurrence and intensity. So the red and the blue box show the differences in intensity, and here this yellow box, which has those 2 peaks that we also saw in the roast coffee, is only visible in the green coffee for Arabica and Liberica, but not for the Canephora. And also for the green coffee, I tried this tentative identification, and I saw that those were mostly possible precursors of the products that we see after the roasting. So furfuryl formates, acetates, and also methanamines, which are affected by roasting. But again, here we have to do compound elucidation with mass spectrometry and reference substances for validation and to really see which compounds are making the difference here. So let's have a look at the ferments for roasted Liberica. And what is visible, and I saw that not only for Liberica but also for Canephora, for example, is that we get a row of peaks, here marked with the yellow box, that is only visible for the ferments and not for the unfermented coffee, roast coffee. That means that with fermentation we are definitely affecting the taste of Liberica or coffee in general. Furthermore, it is visible that in the green coffee, when fermented, we see a significantly lower number of peaks. So we are getting rid of some of the precursor molecules that later on form the taste of Liberica. This could be that we are missing the off flavors or the precursors of the off flavors that were already described by Kenny and Raven with lactic taste or also with the rubber tire taste. So most probably the stuff that we are missing afterwards is the precursors for the off flavors. Now I did a PCA to just see whether the differences are large enough, although if they are visible, they are most probably large enough to really separate those three species with this screening technique. And as you can see here with the blue points here, these are the Arabicas, then here we have a cluster of Canephoras, and here this huge or larger cluster of Libericas, and you can see that here is one yeast that was used for fermentation, there is a second cluster of yeasts that were used for fermentation, there's a third cluster of other yeasts that were used for fermentation, and a cluster of the unfermented Libericas. So we are already able, with just a simple variance-based PCA, to not only differentiate between the species, but also between the yeasts that were used for fermentation. And what I want to stress a little is, if I have a look into the loadings, so the importance of the different peaks that we find in the spectra, is that we see that those peaks that I beforehand marked as visible differences also have an importance in the separation in the PCA. And again, the same for green coffee, and although this doesn't look as beautifully separated, I can still say that you see here a cluster of mostly Canephora, here is a cluster of mostly Arabica, and these three here are just a cluster of the unfermented Liberica, and two of the fermented Libericas just clustered back here. But we need more green Liberica samples, fermented and unfermented, to really validate those results, because this is just like we are thinking that it is possible from those results, but we need more to actually validate this. And again, if we have a look at the loading, so at the importance, you can see that here in the second principal component, there is those two boxes that I marked as visible differences that have an importance, and the other one, like the blue box, is visible for differences in the first and in the second principal component. So these are actually also compounds that we mark as definitely having an influence on the differentiation between the species and the ferments, and with that also in the taste. And this already leads me to my conclusion. So there are differences between the olfactory sense and GC-IMS. I guess that is somewhat clear, and we want to use the separation of aromas in GC-IMS to help identify the differences that trained personnel can taste between those coffees. I compared 3 coffee species and different ferments and saw differences in peak occurrence and intensity, and also, which is especially mentionable, that we have a reduction of the peak number, so the aroma compound number, in green coffee once it is fermented. I did a tentative identification of the aroma compounds, but I hope I stressed enough that this is really just scratching the surface. And at last, I can say that we see a differentiation between the coffee species and also between the coffee ferments in the PCA models. That means that we are able to differentiate between the 3 species with this screening technique, and also between fermented and unfermented coffee with this screening technique. And now for the future, I want to do more of this aroma compound elucidation using mass spectrometry and validate that with the reference substances, as well as try to differentiate not only between Coffea liberica, arabica, and Canephora, but also between Coffea liberica and Coffea excelsa. And I'm currently waiting for some samples from Brazil to differentiate further between microorganisms that are used for fermentation, although those samples are not Liberica but Arabica. And with that, I'd like to thank the Fazendas Dutra and Dr. Schwartz for giving me the opportunity for my stay there and gathering samples there, my working group for always supporting me, the cooperation partners for enabling my PhD thesis, and you for your kind attention.
Thank you so much for your exciting presentation. Probably the first chemical study with so many Liberica samples. Perhaps one quick question from myself. Were you able to identify the classical markers for the species differentiation, like the methyl cathostols, or these compounds? Are these completely different markers you had identified?
So most probably we see the same markers, but the problem is that without the mass spectrometry side, I'm not able to really say that these are the markers. So I need to do the mass spectrometry side as a reference analysis to just say, okay, we see the classical markers, and on top we have those other markers, and then establish the GC-IMS as a screening method for also authentication and quality analysis.
Okay, thank you. Are there any other questions from the audience? I see you are looking forward to the coffee. Yes, there is one. Please go on. There's a microphone coming.
I speak so loud. I think that the question is the origin of the samples, because you are talking about they are from the same region or they are from different regions, where you are talking about the Liberica, the fermented and unfermented, they are the same samples that you analyze before and after the fermentation, because that is important just to see the difference and to establish if there are good chemical markers, yes or not.
Yeah, so we got some that were referenced also for the fermentations, but as I needed to work with what I got in the moment, it's pretty much spread out. So there are some that are references for the fermentations and some that are completely outside of it, so yeah.
But very, very quick answer for me, because I know where the green coffees come from. The Liberica come all exactly from the same field. It's exactly 20 meters on 10 meters, and they have been exactly, absolutely same bin, same soil, same plant, same picker, same day, same. Like the Thai say, same but different. They only have been split into 3 different groups depending on which yeast we used for the fermentation. So that's the only difference. You couldn't make it closer than that. So any university would just love exactly those samples. That's how we made it.
Okay, so thank you